A white blood cell has to swallow a bacterium whole, so it needs a really wide channel. That sentence is the wrong answer to all 10 questions on this topic. The widest pore in any transport protein is 1 nanometer. The bacterium is 1,000. So the cell stops pushing cargo through the membrane. It moves the membrane instead. Here's what a transport protein is for. Glucose, 9/10 of a nanometer, fits. Now put bulk cargo on that axis. LDL, 22 nanometers. A flu virus, 100. The bacterium doesn't fit on the chart. Its bar would run off the side of your screen. And you can't widen the pore. Open it that far and it's a hole, and the cell empties. A pore is narrow because narrow is how it chooses. Width and selectivity are one fact. Watch the sequence. 4 moves. Every one the membrane doing what a wall could not. 1. The cargo settles. 2. The membrane dimples inward until the pocket swallows it. 3. The lips meet behind the cargo and the membrane fuses with itself. 4. It pinches off as a vesicle and the surface closes over behind it. Notice what never happened. No pore opened, no protein flipped it, nothing went through anything. Here's why this second system exists. A pore has a fixed width, a hard size limit. A fold has no width. Bigger cargo just means a deeper pocket. So size stops mattering. 1 mechanism, 3 names. Phagocytosis is the dramatic one, cell eating. Pinocytosis is the quiet one, cell drinking. A tiny vesicle of whatever's floating past. Receptor-mediated is the selective one. Receptors gather one named cargo onto a patch. Same fold every time. Only the contents change. That's all the name is telling you. That's what selecting buys. Drinking gets whatever floated past. This gets one thing, concentrated. Now the sentence that costs marks. There's a protein doing something, so it's transport through a protein. It sounds reasonable. It's the reason this happens at all. And it's still wrong. Split the job in 2. The receptor recognizes and binds. The membrane folds, seals, and buds. The LDL never passes through the receptor. It's held against the patch the membrane folds around. The vesicle is in. So the bacterium is loose in there, floating past your ribosomes? No. Follow the face, not the outline. The side lining that bag once faced the world. Folding inward never turned it inside out. That lumen is a pocket of the outside, carried in a bag. Which is the point. A phagocyte swallows live pathogens and digests them in a sealed room. Only digested fragments cross into the cytosol on a transporter. A broken bag is an infection. Run it backwards and you get exocytosis. Built in the ER, packaged by the Golgi, walked out. The vesicle fuses with the membrane. 2 bilayers merge into one, and a pore opens. The load spills out. Nothing crossed here either. The lumen was always outside. Fusion reconnected it. And the book's balance. Budding in removes membrane, fusing out puts it back. It's recycled. Last trap. No pump anywhere here, so the reflex fires. No pump, so it's free. Wrong. Bending a flat bilayer into a sphere is work. So is forcing 2 membranes to fuse. Topic 2.6 said a protein doesn't mean a bill. This is the other half. No protein isn't a refund. Never through. Never in the cytosol. Never free. Want to step through it yourself? Links below.